I open this academic ceremony in which Mr. Tobias Meßmer will defend the academic thesis Decoding Cultured Meat Production, the transcriptive landscape of bovine satellite cells in proliferation and differentiation. I welcome you and your pioneers, lots of success. I welcome also your supervisors, Professor Post and Dr. Fleck, and of course the people of the opposition who you're going to meet, and all the people and friends and family on site here and those online. But before we start the opposition, I give you time to tell us a little bit in the presentation what your PhD is all about.
Thank you very much, Pro-Rector. Dear Pro-Rector, dear members of the Corona, dear family and friends, colleagues, online viewers, thank you very much for being here today for the defense of my thesis, and welcome to this short presentation. I would like to start by asking you a very simple question. On a warm, sunny, almost summer day like this, what is the first thing that you think about doing? I would take a wild guess and say it's usually not listening to a bioinformatics science talk, but rather something like this. Meeting up with your friends and family, having a nice barbecue. In my family, a good barbecue was just as much part of summer as it was swimming in Lake Constance. And I think if you see that picture, you can almost smell what's going on here. And if you share that feeling, you're not alone. The world nowadays consumes 4 times as much meat as 60 years ago, while we are only 1.5 times as many people. However, you might also feel slightly conflicted about this picture, about eating meat. Maybe you're even vegetarian or vegan despite actually liking the taste, like myself. And that is because, unfortunately, our eating habits are not sustainable. It's estimated that livestock production is responsible for approximately 13.5 of the total greenhouse gas emissions, so it's a strong contributor to global warming. And since it's unlikely that the whole world will suddenly start throwing eggplants and zucchini on their grill, we need more sustainable alternatives to the current meat production system. And one promising solution there is called cultured meat. And when I first heard about cultured meat in approximately 2018, I knew that this is the topic I wanted to do my PhD in. So what is cultured meat? Basically, it's normal meat, so animal muscle for human consumption, but instead of having it produced by the animal, you have it produced outside of the animal using the animal cell. So how do you do this? Well, first you need the cells, which can be isolated from the animal in a first step called cell isolation. Theoretically, many different cell types can be used here. The main focus of in this thesis was on a cell type called satellite cell, which is the natural precursor stem cell for the muscle. So from this isolation procedure, we only get a few cells, but to produce or to eat a lot of meat, we need a lot of cells. Therefore, we need a second step called proliferation. And in this second phase, we let the cells divide. We stimulate their cell cycling so that at one point, if everything goes well, we end up with a lot of cells. However, no one wants to just spoon up cells. We want to actually eat meat. Therefore, we need a third phase called differentiation. And in this differentiation phase, we switch the signals that we give to our cells so to stimulate them in a certain way that they will turn from a stem cell to a functionalized cell. In our case, into muscle tissue. And this muscle tissue you can then have from human consumption. So while this process sounds elegant, there's a reason why we don't have cultured meat on the shelves of our supermarket yet, or rather multiple reasons. As with every new technology, there are technical challenges, but in our case, there are additionally biological challenges. You can see some of them summarized here. So when I first started at Mosameat, almost exactly 4 years ago, I had just finished university, in which in my master's I worked on some projects that involved RNA sequencing. And then I was looking through the literature of the cultured meat field, and it appeared to me that a lot of the problems, like those shown here, that are still existing in this field could really profit from this novel technology. And it seemed like no one was really harvesting this knowledge yet. Therefore, the explicit goal of this thesis was to apply RNA sequencing technology to the issues existing in cultured meat. And just that we're all on the same page, I want to give a very brief introduction to how RNA sequencing works. Don't worry, this will not be very technical. It will just be a conceptual explanation. So you have a number of cells in condition 1 and a number of cells in condition 2. Now what RNA sequencing allows you is to compare these cells in the two different conditions with each other. And what's so powerful about RNA sequencing is that this comparison happens via the gene expression profile, and you don't get information of only one gene or two genes. You get the information about all the genes that you have in your cells. So what does that mean? It means that you can now look at every single gene and you can see whether it's upregulated, whether it stays the same, whether it's downregulated, when you change a certain factor. For example, you have two different time points, or you have cells in two different media. And because the functions of a lot of cells are known, this tells you a lot about those cells. So it could tell you whether the cells might be stressed, which cell type they are, which specific state they are in, and a lot more. And nowadays it gets even more powerful than that, because you can't only do RNA sequencing on a whole bulk of cells, which is why it's called bulk RNA sequencing. You can also do it on single cells. And this really allows you to tackle a whole different range of biological problems, such as asking about the homogeneity of your sample, so how different are the different cells from each other, or population dynamics, or different states of your cells, whether they switch into each other, for example. So I would now love to present every single data set that we created in the past 4 years. However, I went back and counted, and it's actually been 18 bulk RNA sequencing and 3 single-cell RNA sequencing data sets. So there's really no chance of doing that. Instead, I would like to go into 2 chapters in a bit more depth as an example for how we use this technology, RNA sequencing, to decode cultured meat production. So the first problem that we tackled was a really important one to us. If you remember this process that I showed in the beginning, the last phase of this process is called differentiation phase. And when we started, the gold standard, or really the only way to perform this differentiation, required animal components. One that is specifically problematic is called fetal bovine serum, or in short, FBS. You only get this FBS from slaughtering pregnant cows and then taking the blood of their fetuses and give that to your cells. So you can already hear by the sound of that sentence that this is really not compatible with our process. So we wanted to remove it, and how did we approach this problem of removing FBS from the differentiation phase by using RNA sequencing? Well, first, when you have a problem like this, the best way is to try to understand it. So first of all, what we did was we used the standard protocol to start differentiation, which is simply by going from 20% FBS to 2% FBS, which is a process that is called serum starvation. What happens when you do this is, you can see here, the cells will form little muscle fibers that you can see here in green, because we colored it by the protein desmin. We then used RNA sequencing to learn what happens during this differentiation process, and also what happens within our cells during this differentiation process. And we will now only focus on the comparison between day 0 and day 2, because there the transcriptomic difference was the strongest. So this is one result that we got. It's called a volcano plot, because it's a little bit formed like a volcano. It's a typical plot that you would get from RNA sequencing. Here, each point that you see is a gene, and if it's on the right side of the plot, it means that it's upregulated at day 2. If it's on the left side, it means it's downregulated at day 2 or upregulated at day 0. For example, you can see that on the right-hand side, there are 2,743 orange genes. Out of these, approximately 15,000 that are upregulated. Now that's a lot of genes, and of course you can easily get lost in that. Therefore, we have chosen to focus on a few very specific genes, and we focused on genes that code for so-called surface receptors. Now, surface receptors, these are proteins on top of your cells that sense the environment of your cells, and they can get signals from the outside of the cell and pass it down to the inside. And we did that because we hypothesized that if we would now provide ligands to those receptors that are upregulated, it might be possible to stimulate differentiation. So if we stimulate these receptors, to stimulate differentiation without the use of FBS. And indeed, when we started, we started with a base medium that you can see here in light blue called SFB that only kept the cells alive. And then we started adding these ligands to the receptors that we just identified, such as recombinant insulin, transferrin, and both together. And we saw that the fusion index, which is a measure for differentiation, really increased substantially. We then kept adding ligands on top of that formulation, which you can see here, and found that with 10 micromolar LPA, lysophosphatidic acid, it seemed to give the strongest differentiation. And finally, if you compare this now to the differentiation induced by serum starvation, you can see that both the extent of differentiation as well as the underlying gene expression profile in our serum-free formulation are really similar. So in summary, we used RNA sequencing here to identify several surface receptors, then we stimulated those receptors, and we were able to induce differentiation even without FBS. We published this finding in Nature Food approximately 1 year ago, and we got really nice responses from the field, which is why we're really proud of this chapter. So that was one example of how we used bulk RNA sequencing. Now, in the next chapter, we then went on and used single-cell RNA sequencing to dive into a fairly different problem. Meat is a quite complex tissue. There are different cell types in it. Between the muscle fibers, you might also find fat and connective tissues. It has a very defined structure, so our initial motivation was to get closer to this complexity. And to do this, we first had to know a few things. For example, or most and for all, we needed to know which cell types are present in the cow muscle and which of them we can actually isolate and keep alive in the lab. And luckily, single-cell RNA sequencing is just a perfect tool for that. And here you can see our experimental setup. So we first isolated cells straight from a meat sample and performed single-cell RNA sequencing on it. We also kept on growing those cells. So we kept some of those cells and grew them in a serum-free medium. And then after 72 hours, after 3 days, we looked at these cells again and looked which one would grow, which cell types would remain, adhere to the flask. So that means these cells we would actually be able to potentially keep alive in the lab. And then finally, we also purified satellite cells to follow them over time. So the first part is what we got here. So you can see here a so-called UMAP plot. In this plot, every single point that you can see represents a single cell. Cells that have a similar gene expression are clustered closely together, and we colored these different clusters for the different cell types that we identified in there. In the meat sample, we found 11 different cell types, which is also what previous literature has shown about mouse or human muscle. Then we had a look 3 days later, and we found that 3 days later, only 7 out of these 11 cell types would remain. And now, out of these 7 cell types, we were actually able to isolate and culture 5 by designing a very specific antibody panel that enables separating them. So this was really exciting to us because it means that one day we might be able to use all of these 5 cell types to create a multicellular steak. However, we then analyzed the data further and had a fairly unexpected finding that led us towards a more immediate problem. So I told you that we wanted to purify satellite cells. And as to remind you, these are the muscle precursors from which we make the muscle tissue that we eat. And we managed to get them 99% pure. And then we cultured them for a longer period of time and realized that at one point, after 8 passages, their expression profile didn't look like satellite cells anymore, but a lot more like a different cell type called FAPs, fibroadipogenic progenitors. And therefore, we concluded that these 1% of fibroadipogenic progenitors that were there in the beginning of the culture must have overgrown our cells, or to say it differently, that the conditions in which we grew our cells in were much better suited to the FAPs than to the satellite cells. Consequently, we went back to the data to figure out what was different between those two cell types, and we tailored the growth conditions to account for these differences. And by this, we were then able to create a new medium in which the satellite cells would be much better able to grow, which is why nowadays we're able to keep our cultures of satellite cells pure for pretty much as long as we want. So these were just 2 examples of how we use RNA sequencing in this thesis. And I hope I was able to convince you that it's a really powerful tool for cultured meat research if we're asking the right questions. So with that, I would like to give the word back to the prorector.
Thank you very much for a clear presentation. Then we will start the opposition now, and the first opponent is also the chair of the assessment committee. That's Professor Slimer, and she has also accepted the role of secretary today, for which I'm very grateful. And she's a professor in pathophysiology in KRM in the University of Maastricht. Professor Slimer.
Thank you, Prorector. Dear candidates, it's a pleasure to be here, even though it is sunny, and the barbecue would, of course, always be good. But maybe in future we can have a barbecue with Mosa meat on it. So for today, of course, we're discussing your thesis, and people always say it, but I did really enjoy reading it. It was quite outside my own box, and it was absolutely very nice. Most of it I enjoyed the methodological way you approach the things, the 4 things you just described, and you take it much further than many people with single-cell sequencing do, because they just describe things and you take the data and you intervene afterwards and solve a problem. So really very nice, and congratulations, of course, also to both your supervisors. So before we dig into things very deeply, I was wondering, since you also had such excellent graphical designs, whether you can highlight a little bit more detail on your cover.
Interesting. Oh, dear highly esteemed opponent, thank you very much for these nice words. It's really nice to hear. And also thank you for the nice opening question. So the cover was a bit to represent this transcriptional landscape of the meat. So this whole problem that we are facing is kind of this huge mountain, and when you stand in front of it, it might look scary because it's so big, and you think that there's so many problems that you have to overcome. But once you start really mapping it out and start walking up, it becomes a bit easier. And that's a bit what the cover should represent, that you...
I figured there must be more to it than just representing a landscape. So very nice, very nicely done. So back to the science of things. You choose single-cell sequencing, but you're also very interested in cell state transitions. So quite often, people use another tool called ATAC-seq for that, because it's, in my opinion, it would more be able to tell you a little bit more specifically about what gene is activated at this certain time point in the transition. So I was wondering, were you consciously choosing against ATAC, or just weren't familiar with the technology at the beginning, or is there any other reason?
Thank you very much for the question. So when starting 4 years ago, the first plan was to actually do a multi-omics approach, and in addition to RNA sequencing, also go into ATAC-seq and more genome sequencing as well, and explore really not just the transcriptome, but also the epigenetics and the genetics. And ATAC-seq is a perfect tool to also assess the epigenetics to see which areas could be are open to transcribe. And the main reason why we didn't do that was because we already had, so the cell state, the cell states came up quite late as a result of our RNA sequencing findings, because we first were interested in how So in muscle, you have a high heterogeneity of satellite cells. Some of them might be quiescent, some of them might be active, and we thought this heterogeneity might also be captured in vitro somehow. So this is why we use single-cell RNA sequencing first on it, and I fully agree that it would be very exciting to follow up on this, but it's also, then you also have to find collaboration partners to, again, work on this, and the timing was not yet there, but this is certainly something I would be excited to work on.
All right, that's a good answer. So in line with this, in chapter 3, you're trying to study, like you just showed, to prevent the overgrowth of these unwanted cells, the fiber adipogenic progenitors of FAPs. And you quite quickly come to the conclusion it's that you sorted them there, and that's why they're there, and they just take over because they're better proliferators. But if I look at our studies and studies in the field, cells change their transcriptome quite a lot when they hit the plastic, or even if they don't go exactly on plastic. And a lot of cells also in nature, in pathology more so than in physiology, they transdifferentiate, so they become another cell type. So in your opinion, what are the chances that maybe your satellite cells actually become more FAPs because they are in a different environment?
Mm-hmm. Great question. We also thought about this a lot, to be honest. When we first saw that problem, that we would suddenly have a completely different phenotype of the cell, because we didn't only see that in silico, so in our computational data, we also saw it in the well, because they would just behave very differently. For example, if you give them, which you can also see, I think, in figure 3 of that chapter, 4, you would. When you would stimulate differentiation, for example, they would behave very differently. So they would not show any behavior that we know from satellite cells. The main reason why we thought it's not transdifferentiation was because we saw two very distinct, clear states or cell types. We never saw any smear or transition between those. So we assumed, and we also saw that not just in our in silico data, but also when we performed FACS on it, because we had good, reliable antibodies for both the satellite cells and the FAPs. And when we would check them on different times, we would never see, like we would always see two very clear populations and no intermediate population. And there would have been some likelihood that there's some in-between state, and we've never seen it.
I would challenge you to do some pseudotime analysis on the whole set, not just in figure 5 on the satellite cells, but it's good enough evidence for the moment. Thank you very much. I give the word back to the prolector.
Thank you very much, Professor Sluimer. Then we come to the following opponent, who's also a member of the assessment committee, and that's Professor Evelo, and he's Professor of Bioinformatics for Integrated Systems Biology at FHML and the Department of Bioinformatics of the University of Maastricht. Professor Evelo.
Thank you, Mr. Prorector. Dear candidate, I was impressed. So let me congratulate you first on this thesis and the impressive work that was in there, because I really was impressed. And let me extend that congratulations already to the whole team. And I was a bit sad too. I thought like, wow, this is happening just around the corner. I wasn't really aware. I was aware of the kind of things you work on, but not the kind of approaches you had. And I thought we should have talked way earlier. So let's try to do that now. And I want to first quickly, I hope, move to your table on page 131, which mentions, you don't even need to go there, I think, but you mention all these breeds of animals that you get the samples from. And I was like, hey, I may be a bioinformatician, but that thing has happened to me before. So I was wondering, can you briefly explain me why you actually wanted to look at different breeds? And then I will tell you why we looked at it before and see whether we can go from there.
Highly esteemed opponent. Thank you very much for the question. And again, thank you very much for the really nice words. Indeed, we should have probably collaborated earlier. So we chose different breeds to see if there would be some trend, at least, in their phenotypic behavior. The breeds question has always been very high up on our minds. It's the first intuitive thought that you would, or the first factor that you would consider when you think of how might satellite cells behave differently. Because potentially you could, if you just look at different cows, if you look at a Belgian blue cow that is just pure muscle, and next to it is a Limousin milk cow or so, you can see that they're phenotypically extremely different. So the first thought that comes up is that also represented in vitro? And our main data, or our data that we now have collected from over 270 different animals, rather points in the direction that there's no strong correlation, at least, with the breed, which was fairly surprising to us. And this is also why we chose to use different ones in this chapter, because we thought that it will not play too dramatically into account there. But we were still interested to see whether.
So let me take it from there, because I think I know what's going on. At least my collaborators from Iran said something different. They say, hey, if I just look at the meat of these animals, it's indeed different. It's not just the whole animal, it's also the meat. And they said there's a process going on in some of these breeds that is called marbling, by butchers at least, right? And that shows that there is bands of fat tissue being formed. And now we're getting close to this co-culture cell things where you were trying to keep cells out, but she was trying to get cells in, right? And then I'm wondering, and then I want to go through this, well, on a kind of a geological trip to watch volcanoes. Then I'm wondering, if you want to look at that type of complex processes, do you still think you can just take, like you did for the volcano plot, like we focus on a few genes that actually are on the outside of the cell, or do you think we should go deeper into the cell?
I fully agree that we should go deeper on also studying the genetic background of the cells, because of course what RNA sequencing gives you is insights into the gene expression, so you get hints from where these differences might come from, but you don't really, it's hard to discern cause and effect. You might just see some different genes being transcribed, but you don't really know the reasons why. When you would, for example, now use whole exome or whole genome sequencing to find really differences between breeds and then try to correlate that to the behavior of your cells, you would either need a very strong phenotype, or very strong differences between, let's say, breeds, or you would need smaller differences, but then a lot of animals. And now we approach the...
Or a lot of genes.
Pardon?
Or a lot of genes, which you
actually
have,
go very
right?
deep in it, and then you would need to perform whole exome on like 10, 20 different animals. And that itself might already be almost a PhD project for itself, because it would be a fairly complicated study. So that's why we didn't really approach it, and the main reason for not approaching it was that the differences that we saw were just not too strong. If I can come back to the co-culture, there it is true that, for example, we see when we take samples from younger animals that we get, for example, a bit more satellite cells compared to the FAPs. So there might be a trend there, which is very relevant in vivo, because there, of course, the different ratios matter more, but we perform the proliferation and the differentiation phases separate from each other. So when we make our product in the end, we mix the different tissues after they've already been differentiated. So these these effects are a bit less relevant, because you don't have to worry about, while you perform differentiation, will I have the amount of FAPs that are sufficient to get me a nice marbling, because we create that after the differentiation process. Because performing co-differentiation would require even more optimization. And that would require creating, for example, a medium that allows the differentiation of two cell types in parallel.
I think what you're showing is that we really should have started talking earlier, because I tend to want to challenge the thing you tell about cells and then go to the volcano plot and see how you better can treat the genes and use more of them and actually find paths down from that volcano again. Mm-hmm. But I think I have to give the word back to the pro-rector and leave it at this for now.
Thank you.
Thank you very much for your position, Professor Evelo. Then we come to the following opponent, who's online, and also a member of the assessment committee. That's Professor Henkel. He's professor in cellular agriculture at the Technical University of München. And as I said, he's online. And thank you for coming all the way from München, Professor Henkel. We are always glad with guests from outside, albeit online. The floor is yours.
Thank you very much, Pro-Rector. Thanks for inviting me. Thank you also for your excellent thesis that I was able to read, like not so many weeks ago. I am, by what you told me, a little bit reminded back to my own PhD thesis, where I was intensively looking for bacterial subpopulations. I was doing performance studies on individual bacteria, and I got at the same point where you arrived at this conclusion that there are maybe not the population that you want, or maybe not the things that are happening that you want. So this was an issue for me as well. So I think this is something very, very important that we have to be addressing for the future for the cultivated meat production. And myself and also my team, we are focusing heavily on application. We are focusing on how we can use that basic knowledge that you developed for actual application purpose. And this is why I would like to ask you, what do you see the potential of these findings for actually driving, let's say, a perfusion process where you intend to have the desired marbling in the end? How do you see the application perspectives for this? And more precisely, how would you see the perspectives of actually online determining of what is going on inside your whole 3D construct that you will probably be looking at?
The highly esteemed opponent who's online, thank you very much for this great question. So if I understand your question correctly, you're asking about how we can apply RNA sequencing to be a bit more process-directed. And what I want to point out there, that it's a very exciting technique, but it's by far not the only and not the only solution to every problem that is out there. And I hope I didn't give off the impression that I think that, because there are some limitations to it. For example, what you can do with RNA sequencing that I think you could also very nicely apply to really production is you could find out what, for example, if your cells are stressed, so you put them in a bioreactor environment and they are exposed to shear stress and to sparging stress, you could come up with signatures, or you could find out what genes, for example, they express once they get stressed beyond a certain threshold in a upscaled environment, and then you could use that information to find markers to monitor them. So you could, for example, see that some surface gene is really upregulated. We hope that CD151 might be one of those. Turned out that it was not really the case, but you could find markers through RNA sequencing and then use this to monitor your process. So you could imagine that you have a marker, let's call it a stemness marker, that you can see goes up over a bioreactor period of 30 days. That corresponds to 25 population doublings. And you can see that it goes down at the time when your differentiation would also go down. Then you could, and you would have a marker for that. Then you could regularly take samples of your cells, and you could see if that marker starts coming up. Then it's time to harvest. So you could really have this continuous feedback system that is informed by RNA sequencing. I do not think, though, that RNA sequencing might ever be process suitable beyond the R&D point of view, because it's just too much. You have to put too much thought into the experimental design, and there are different constraints that come to the sample preparation. You have to be very careful to not get any RNases in your samples, and I think for actually having it applied in the process on the go, it might not be the perfect tool. I hope that answers your question.
Yeah, thank you very much. Of course, it's usually a much bigger challenge than we know from standard bioprocess engineering, where we have, like, say, sensors readily available. Now for the tissue cultures, this is much more of an issue. Now, for me, you have a lot of data. You generate a lot of data. The outcome of the experiments is pretty much well documented, and we have a good or sort of good understanding of what we want the outcome to be. So let's say we want a certain degree of marbling, as you told us as well. From my perspective, next to doing a lot of applications in these reactor systems, we really like artificial intelligence, using that for complex problems. Do you see any way, or have you planned for the future somehow to take this complex data set and integrate it in a way that an AI could predict how the marbling pattern will look like depending on what conditions you set?
Thank you very much for the question. Not me personally, to be honest, but we have at Mozambique a very active bioinformatics group, and they are very keen on putting all the data, because it's not just been these 21 data sets from my PhD now, it's also, I think by now, over 100 other RNA sequencing data sets that we've created. So there's really a lot of, and also other bioinformatic data sets, so there's really a lot of knowledge there. And of course, it makes sense trying to find the overarching, the overlapping factors that combine these. Maybe you can see that you sometimes use specific cells from a specific animal, and you can see a trend there that is not obvious in 1 or 2 experiments, but it will be obvious in 20 different ones. And if you can train a model where you can feed in all this data, and then you have it analyze certain outcomes like marbling, although I foresee that to be a bit more difficult because that's more product design. But other behaviors, then I certainly foresee AI to have a very important role there.
So thank you very much. I will be in touch about this because I have an idea how this might be developed for the future. So thanks. My time is up already. I would like to give back to the prorector. Thank you.
Thank you, Professor Henkel, for your position. Then we come to the following opponent, who's also a member of the assessment committee. This is Dr. Snijders. And Dr. Snijders is assistant professor in bioinformatics at Human Biology Movement Science in Nutrium.
Okay, thank you very much. I'm not bioinformatics at all, so basically human muscle physiologist. That's what I study. So, but thank you very much, prorector, for that. So I would also like to start, dear candidate, to congratulate you on the work that you've done. I think it's very impressive amount of work in well-designed studies that you have been performed really on the, I have to say, cutting edge of science, on progressing towards like creating this cultivated meat, which I think is really exciting time to do research, and I think it's also very much a promising future in that, and I would like to extend those words also to your supervisors, of course. So as I said, I work as a human muscle physiologist, so a lot of the techniques I'm only jealous about, like what you can do and how you are able to investigate certain pathways and how cells really work. But as part of your dissertation, you also have to have something about like an impact of like your science and your studies that you have done in a larger field of other scientific research fields and societal fields. And I would like to have a short discussion on it, and I would like to invite one of your assistants, because I always like to get them involved as well, to read just a single sentence, so on page 199, and I will tell you what sentence to read, and I would like to have a short discussion on you with that. So, don't matter who, but it's on page 199, it starts at the top. The second sentence of the first paragraph, it starts with, although there's only limited. Can you read that sentence for me?
Yes, 1 second.
Yeah, sure.
(199).
Yep, page 199, and it starts all the way at the top, the second sentence, although there's only limited transferability.
Although there is only limited transferability of results from cells cultured outside an animal to their natural environment, the detailed characterization of satellite cells in this work can also contribute to a better understanding of skeletal muscle biology necessary for treating muscle-related syndromes such as sarcopenia or Duchenne muscular dystrophy.
Thank you very much. Really good. So, so I also study satellite cells, also in relation towards aging. So I think you know what type of question you're going to get from me. So I really hope that I can learn something here today. So, because you mentioned that there's also maybe sometimes difficult, but there's some transferability maybe to understand also how satellite cells behave in vivo and maybe also in relation to sarcopenia. So my open question is, so what is it that I need to know from your work that I can use basically in my day-to-day science understanding on maybe improving satellite cell function in vivo as well?
Dear esteemed opponent, thank you very much for that terrific question. And it's actually a very exciting discussion to have, and I think 5 minutes will be very difficult for this. So I was surprised by how little information there is out there about in vitro aging of satellite cells, because most of the studies, and if I may give back the compliment, because I also read your thesis quite a bit in preparation of for this.
That's
thesis.
Always good. That's right.
And so there's really a lot of information on that, for example, compare young satellite cells from young donors to satellite cells from old donors. So there's a lot of differences there. And one huge difference compared in vitro to in vivo is that in vivo we have this extremely complex environment. We have the bloodstream that is different. We have the whole different cellular environment that becomes different. We have maybe some fibrotic tissues that start in the muscle, and all of these have an impact on satellite cells. Of course, they have to have that. And there are also some studies that see that if you take out those old cells and put them in a different and younger environment, that will also change the satellite cells again. So it's really clear that this extracellular environment has this huge influence. Now, we're in an in vitro environment, so one of the few things we can really control fairly well is this extracellular environment. So all the changes that happen to the satellite cells in that time are most likely attributed to processes that happen inside of the satellite cell. Now, it's of course difficult to assume that what happens inside of the cell, if you just ignore the outside, in vitro and in vivo is exactly the same, but there are certainly overlapping features. We have explored some of them, like the transcriptomic ones. Then there's certainly some artifacts, like for example, in vitro you can also see that they become become bigger during a longer period of culture, which I don't think the data is on in vivo, that satellite cells from older animals would be bigger than the ones from younger. So there are artifacts, but there's also comparable things. And these are, for example, things like telomere shortening that you can see in both, or like ROS accumulation. And so
because something you see like in one of the chapters as well, you talk about like this aging of the cells, that they do not proliferate at some point anymore, and that's a major limitation of like going towards like producing cultured meat in the end. So what is then that, because you say something that there's also that some of these cultures that are like exhausting or like exhausted, they don't proliferate anymore, but they do show different gene expression at the beginning compared to other cell types. Is there anything that we can learn from that maybe can predict sometimes, or that can have an impact on like these cells, how they are able to behave themselves, and whether that can also be something that translates towards humans?
Yeah, so the data that you're referring to is we cultured the cells over a long period of time, and then we saw the differences. Some of them would stop growing, some would then continue growing. And then that was at passage 6, and at passage 2 we compared them already, and we saw some slight differences there in RNA sequencing. I want to acknowledge that these differences were not dramatic, so there were hints, and we didn't have the time to yet really confirm all of them, but there was a higher expression of myogenic-related genes in those that would stop proliferating at a certain point. So a potential hypothesis there could be that if your satellite cells exhaust too much, so if they regularly enter this committed or like almost differentiated state, they would not be able to proliferate as or to replicate as much anymore. So you kind of exhaust the pool, and that is definitely a concept that people think of in vivo as well. But I wouldn't go so far to say that you can really already draw firm conclusions for the human. So maybe for the cow, but also there might be species differences. But just concluding from in vitro to in vivo will always have certain big limitations.
Okay.
And yeah.
Thank you. Do we have more time for one more question? No, that's it. Oh, yeah. Thank you very much. I'm happy with your answer. Thank you.
Thank you, Dr. Snijdersen. Sorry for calling you bioinformatics. That's the next opponent. We have so many affiliations. Dr. DECODING, he is bioinformatics at clinical genetics at the Maastricht University Medical Center. Dr. DECODING.
Dear Prorector, thank you for giving me this excellent opportunity to exchange some thoughts with the candidate. Dear candidate, I want to congratulate you with the wonderful work that you did, and which produced this amazing booklet. It's called the transcriptomic landscape, and that's, well, as you know, the second big element in our central dogma of molecular biology. I'm working in the laboratory of clinical genetics, and therefore we like to start at the beginning, the first bit, which is the genome. So while reading your manuscript, I was thinking about that. I wanted to ask you why you didn't do any experiments on the genomics of skill. But then I hit this little chapter in chapter 4 about copy number variations, and then I thought, wow, excellent opportunity to ask some questions about it. And I wanted to draw your attention to the picture on page 125, in which you showed the results about from the CNV analysis. And my first question basically is, what were you trying to solve in this experiment?
Dear esteemed opponent, thank you very much for the question. I would say solve is the wrong word to what we try to do there. We try to understand what was happening to our cells over this long period of time, because we knew that they would change. So we wanted to find out whether these changes come from genetic variation. And you have different tools, some of them more fancy than others, and to dive into this genetic variation. And one of the most common genetic variations is structural variation. So you have parts of the chromosome that end up somewhere else. You have maybe some deletions of bigger structures somewhere. And what we were interested in this specific experiment was whether, when you culture them over time, because this is also something that has been described in literature quite a lot, where you culture them for a long time, you will start seeing genetic structural variations everywhere, and then on top of that, whether you might be able to correlate those differences with the behavior that we see, because we saw 3 very clear phenotypes. We saw cells dying, then we saw cells continue to proliferate, and of these that continue to proliferate, some would still be able to differentiate and some were not. So we were thinking maybe we find one phenotype there where we can see, okay, this region is deleted for all of these genotypes with this certain phenotype. So we were hoping that maybe we would get a clue that some genes in that region might be important to this behavior. Unfortunately, there was no such correlation.
Thank you for that excellent explanation, because actually I was hoping that you would tell something like that. Because what you did see were actually a number of occurring CNVs. Mm-hmm. A driving force behind CNVs is random mutagenesis. So why did you expect to be able to correlate a distinct type of CNV to the broad phenotypes that you see?
My hope would have been to do this experiment with whole exome sequencing, to be honest, but we chose the method that was available to us because we couldn't find the right collaboration to do a whole exome sequencing, and to be honest, we also didn't put quite as much money, or we couldn't locate as much money as how it would have cost to perform this particular experiment. We would have probably created a different experimental design if we wanted to go in with whole exome sequencing. So
sorry to interrupt, because with the whole exome sequencing, you would even have found
SNPs.
Even more distinct changes.
Yes, but
I was just wondering why you choose to look for similarities between your phenotypes and not try to explain the differences that you saw and try to correlate those differences to the phenotypes.
Well, we try to find the differences between the different phenotypes, right? Via the CNV analysis. So once we see differences in behavior and we see differences in the genome, then you can really start to see correlations there. Not necessarily causations, but you can at least assume that some of these regions could be responsible for the behavior that you see. And that's why we chose that.
Because the phenotype that you were studying,
and
I have to end this, I think, but are very broad. I mean, proliferation is a main activity of every living cell. So the underlying processes, they're broadly ranged among the whole genome. So that you see actually differences might not mean that there's no phenotypic cause.
That is true, yeah.
It could also just be a different mistake being introduced, but that gives the same phenotype.
Yes, I fully agree. It would never give you a clear answer. It would only give you a rough idea of what to study next and what to go deeper into. But I think it's the same with if you would have really located some single nucleotide polymorphisms that you can say, okay, all of our genotypes with that behavior show exactly that one. You still can't be sure that this is the reason. You still have to dive into more functional analyses afterwards.
Thank you for your answer.
Thank you.
Thank you, Dr. DECODING, for your position. Then we come to the following opponent. It's Dr. Baker. And Dr. Baker is Assistant Professor in Materials Science at Merlin University of Maastricht.
Thank you, Mr. Prorector, and dear candidate, as said by several excellent theses, and it was a pleasure to read, though I am not an RNA sequencing expert. Before I dig into, I want to see if we can make a few agreements, or if we can agree to a few things, if that would be okay with you.
The esteemed opponent, absolutely.
Beautiful. So there's been a lot of talk today about landscape and environment, and you have a beautiful cover talking about your landscape, and what I have to point out is maybe not a two-dimensional, but a three-dimensional surface. We talk about barbecues. Barbecuing in the rain is not so much fun, is it?
Agreed. Well, you can barbecue at any time, in my opinion, but I might.
Fair enough. And I dare say that your environment can affect what you do. And I dare say that I want to see if we can agree. You might even be a product of your own environment, where the MOSA gave you the chance to do the PhD. Your support of your family and friends have maybe even led to this moment. Would you agree to that?
Oh, I fully agree with that. I think I wouldn't be here without any one of the people here.
So if we were just to look at you and what's inside you, we might miss a lot of what is going on that makes you behave the way that you do. And you can see where the questioning's going. So a lot of your experimental results has been done on two-dimensional surfaces, when in fact the real environment of muscle is a three-dimensional animal. Now, that's a problem, and you're trying to fix this, and we'll get to that in a little bit. But how much do you think, for example, you look at how surface receptors change during differentiation, how much do you think that 2D environment is reflective of surface receptor changing in 3D environments as well? How far off do you think we are?
Dear esteemed opponent, thank you very much for this great question. So I do fully agree that 2D and 3D environments are highly different, two highly different fields. So I would say during proliferation, this is currently in our current system still yet a bit, so not as grave, because it's more a 2.5 dimensional environment, because we still grow, our cells are still attached to microcarriers,
right,
coated microcarriers, so there's certainly some transferability there.
Right.
For the 3D environment, it's certainly different, which is why it was really important to us to confirm results that we would see, for example, in chapter 2 and in chapter 5. Chapter 2, where we created the differentiation medium,
yeah,
chapter 5, where we try to extend or maintain the stemness
right,
better with this component, to extend the findings not just from 2D to 3D to see if they replicate there.
Yeah,
but the understanding part is a whole different question. So what would be interesting, for example, is not just to perform RNA-seq on 2D differentiation, but also on 3D differentiation. We tried that. We had technical problems, let's say, because once you have to digest
We'll come back to those technical problems in a second, if it's okay. So I thought it was actually really extremely cool that you took that step. So you took that step of, all right, we optimize this in 2D, but then especially in chapter 2, let's go to the 3D environment and let's see if it works. So you added a certain number of small molecules to your culture medium. You do so in a 3D environment, which is now collagen and matrigel. I won't talk about matrigel, but it has its own bag of problems. And it does work, right? Mm-hmm. But then in chapter 5, all of a sudden it doesn't work, right? You, if I can have one of your paranymphs read Stelling number 7 quickly for us.
Maintenance of stemness through p38 alpha beta MAP kinase inhibition can be hampered by coating-dependent off-target effects.
Thank you. So off-target effects is often a keyword for we have no clue what's going on. And now you have a beautiful example in chapter 2 where 2D translates to 3D nicely, and in chapter 5 you have a beautiful example of where it does not at all. Mm-hmm. What do you think is going on there? What would you call off-target effects if you had to spend a few minutes talking about them?
Yes. So this was one of the... So this was a project from this year, or last year actually, and it was one of the most exciting problems that we had there, because we see this very clear improvement when we add this p38 inhibitor. Then we go to 3D, it doesn't work anymore. And we were completely surprised by what was the reason for that. And then we looked at what was different there, and we found that for different reasons that I don't want to go into now, these microcarriers that we have, these Cytodex-1 microcarriers, they're coated with vitronectin instead of laminin, and we used laminin on all our studies. And and we wanted to be consistent, so we always used laminin. And then we realized the microcarriers are coated with vitronectin. So then we thought, okay, if the coating is different, it's legit assumption, hypothesis that this might be the reason for these differences. So what we did was we went back to 2D, and then we coated there with vitronectin, and we saw that the p38 inhibitor also didn't work anymore on 2D because we now had this different coating. But luckily, we had different p38 inhibitors as well, not just SB20, 35, 80.
Let me ask you a slightly more specific question along the way. Do you think that the coding affects the pathway as well? Or do you think the coding has no effect whatsoever on the pathway that you're trying to inhibit from the receptor?
I think it's almost it must is a strong word, but we're fairly certain that it has an effect on it, because we saw that once we have this different coating, the responses would be completely different, although...
You may finish your answer. You can also say enough is enough.
I would like to quickly...
Good.
Okay. Thank you very much. So we think that the difference that we saw, we are quite certain about this difference because we, once we coated with the different one, we saw that the effect would go back. So it has to be an effect from the coating. And we also saw that the p38 inhibition still worked. So we looked in Western blots, and we saw that the inhibition still worked. So this is why we call it off-target effects, because it must go through some different pathway. And this is likely to be affected by the coating as well. Okay, thank you.
Thank you, Dr. Baker, for your position. And Mr. Candidate, the time appointed for the defense of your thesis has now passed, and the committee will now withdraw to discuss the quality of your thesis and of your defense, of course. And I request that you and your company on site and online await the results of our deliberation and our return in this room. And with that, I suspend the meeting.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
I reopen this academic session, and Mr. Meßmer, the degree committee here present has assessed the quality of your thesis and your defense, and in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor, and Professor Post is authorized to confer upon you this academic distinction in accordance with Dutch university custom, and invite your supervisor to now take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Great. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Tobias Meßmer, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. I'm a little bit handicapped here, so... Congratulations. I would love to give a long story and a long kind of other congratulations, but Dr. Meßmer, congratulations, and also congratulations to your family and friends. But Josh is actually dying to give the laudatio for you, so I will give the floor to Josh.
Testing. Thanks, Mark. Now you're properly nervous, right? So, yeah, congratulations, esteemed candidate, or should I say now, Dr. Meßmer. I just wanted to give a little bit of background, actually. Slightly unusually, I knew I was going to be Toby's supervisor quite a bit before we actually met, and I'd already heard a bunch of good things about you from people you'd worked with in the physiology department, experiments that you'd already performed during your internship at Mozambique, and I knew you'd already contributed quite a bit to a paper. And yeah, before you even started, you were firing me multi-page emails, all sorts of different ideas for experiments we could try, methods we should be testing, and yeah, it was obvious that you were super keen to get started, and yeah, the expectations from Mark and I were pretty high, to be honest. And yeah, needless to say, of course, when you eventually arrived, those expectations were not just met, but really far exceeded. Judith and some of the other guys on your committee have already kind of commented on the fact that getting a PhD is about becoming an independent scientist. And for you, that was never really, let me adjust my page. That was never really a question, to be honest. From day one, you were already equipped with the skills that you needed to push the boundaries of human knowledge. We've heard today about your bioinformatic approach to cultured meat, and that was never really something that Mark and I had to guide you with or help you with. That was really your own concept, your own project, and you took that and ran with it to massive success, as we've heard about today. It's never felt like supervising you as much as giving you some people to bounce ideas off, perhaps show you some new areas that you could look into, and of course, making sure all of your figures were properly aligned, something which took up an unusual amount of our time. It's really a testament to how much work you've put in, that plenty of the projects you've worked on didn't even make it into your thesis or into the presentation that you talked about today. Yeah, you probably had enough to fill 2 or 3 more theses full of RNA-seq data sets, and all of that amidst a global pandemic as well. But yeah, I think what's really cool, and what Judith also mentioned, was that you didn't just stop at the bioinformatics, as lots of those types of projects can be bound to, but you really delved in deep and found interventions that help us with our actual process. So yeah, your work's really helped to set the research direction at Mozambique. As you mentioned, that's to the extent that we now have a growing team of bioinformaticians who work with scientists to sort of enact the exact kind of vision that you helped to set. Alongside your PhD, you've also stuck to your principles. You've helped to share knowledge and expand the cultured meat field. That's something that's always important as scientists and as a PhD, but in our kind of new and burgeoning field is even more important. You've given excellent presentations at various meetings all across Europe. You've helped co-organize the Cultured Meat Symposium here in Maastricht, and you're often disappearing off to these lengthy meetings of the CELAG Germany Society that apparently you helped co-found, although I don't think anyone else has ever heard of this. You've been a great mentor to junior members of the lab and to interns who've worked in the cell biology team. And yeah, in the last year, it's been really awesome to see you stepping up, brushing up on your project management skills and your people skills to help lead our satellite cell proliferation project. And all of that while managing to come up with about 10 or 15 cups of tea a day for the whole office. Of course, it wasn't all just hard work. It's been a great pleasure to get to know you more personally as well, outside of the lab and outside of the office. You've been at the center of the team's social scene since the beginning. You taught me that Germans can actually, in fact, have a sense of humor. And we've had plenty of adventures, ranging from late-night handboarding, dodging avalanches in the Alps, all the way to the famous, or maybe infamous, techno bunker parties hosted in your cellar. And I think the whole team especially enjoys your choice of team socials. Watching you do laps around the wakeboard park while the rest of us queued up to faceplant into the lake was really a peak moment for all of us, I think. And yeah, along the way, it's been awesome to meet your family and friends, some of whom are here today to share this moment with you. So yeah, I think that about wraps it up. Yeah, even under contractual obligation, there's only so many nice things I can find to say about you. So yeah, we're really lucky to have you here at Mozambique. I know I speak for Mark and everyone else when I give you a massive, massive congratulations. We're all super pleased that you'll be staying to keep working with us beyond this week. And yeah, I think we're all looking forward to celebrating with you this afternoon. So yeah, congratulations, Dr. Meßmer.
Dear Dr. Meßmer, also on behalf of the Maastricht University and the deans, I congratulate you with the degree you have acquired. And I congratulate, of course, also Professor Post and Dr. Vlak, your supervisor, with this success, and your family and friends. And I hope that you will celebrate this in a great way today and perhaps later also. And of course, I wish you lots of success with your new degree in your path on your further career. Having said that, I thank the members of your position for their work, and I thank Luc Peters and Joshua Vervelu for their technical support so that this hybrid session was possible, and of course, the BIDO. Having said that, I'm almost going to close this session. I'm going to explain you what's going to happen. I'm going to ask the guests here to leave the room and go to the rafter where the reception is. We will stay here and make a picture with not the candidate, the young doctor, and Professor Helmer, and then we'll make a picture at the top of the foot of the stairs, so the photographer can stay, please. And then we will enter the rafter, the reception room as well. And if you don't know when you then have the opportunity to congratulate the candidate, it's quite simple. We go first and then you go. And so timing is simple. Yeah? Having said that, I close this meeting.
Please can also be